Assalamu alaikum wa rahmatullah. I am Assistant Lecturer Lamia Hadi. Our lab today, I will talk about somitogenesis, the first lab in the second semesters of the embryology curriculum. The first slide, 18 hours in the chick embryo. In this slide show the bilaminar germ disk and the trilaminar germ disk. Initially, during the second week, the embryoblast cells form two layers. The epiblast, columnar cells in the shape, and the hypoblast, cuboidal cells in the shape. Two layers, the epiblast and the hypoblast, that the bilaminar germ disk. The bilaminar germ disk consists of the epiblast, columnar cells in the shape, and the hypoblast, cuboidal cells in the shape. More development show grooves. This grooves is called the primitive streak, and cells of the epiblast, columnar in the shape, invaginated into the grooves, changing their shape from columnar to the flask shape, were invaginated or invaginations in this process, which called gastrulations. The invaginated cells then migrate lateral to form a mesoderm in the middle, and endoderm after being intercalated with the hypoblast, and the epiblast form ectoderm. The lateral show ectoderm, mesoderm, and the endoderm. This the trilaminar germ disk. Trilaminar germ disk. The trilaminar germ disk consists of the ectoderm, mesoderm, and the endoderm. Next slide. 33 hours in the chick embryo. In this slide show a more advanced stage of the development of the chick embryo. The ectoderm is thickened, forming a neural plate, and also you can see the mesoderm and the endoderm. Development show organogenesis is beginning. The neural plate is folded, forming a neural fold surrounded a neural groove, and the ventral, the neural fold, a notochord. A notochord formations by the invaginating epiblast cells at the primitive pit, separated anteriorly in the midline, and lateral forming a soft tissue, that the notochord. Also, in this section show the mesoderm is divided into الباراexal mesoderm on the side of the notochord and neural fold. الباراexal mesoderm later forming somite, and intermediate mesoderm which is later forming the kidney, and a lateral plate mesoderm divided by a small cavity. This cavity is called the intraembryonic cavity, and a lateral plate, 2 layers of the lateral plates, the somitic or parietal lateral mesoderm adjacent to the ectoderm, and the visceral or splanchnic layers adjacent to the endoderm. Also in these sections show the cell at the tip of the neural fold in that side, and in that side are the neural crest cells. It's part of the structures, formations, the skeletal systems, locations below the ectoderm, on the side and on the side. It's a part of the ectoderm. A neural crest give rise to many structures and many organs. And more development, the neural fold closed for forming a neural tube, and you can see the regions of the neural crest and the notochord. And the paraxial mesoderm is more compact mass, and that is formed mature somite. Mature somite containing to the in the central containing to the cavity. This cavity is called somitocele. And show the intermediate mesoderm and the lateral plate of the mesoderm, 2 layers, the parietal layers and the visceral layers, and we can see the cavity between them is the intraembryonic cavity. In next slide, transverse sections 72 hours in the chick embryo. And these sections show more differentiated of the somite from the paraxial mesoderm. During the 4th week, each of the somites has 2 parts. The ventral and the middle part of the somite, the loose tissue mesenchyme around a neural tube and a notochord is formed sclerotomes. This mesenchymal tissue differentiated later, forming a fibroblast, chondroblast and the osteoblast of the vertebral columnar. While a dorsal and the lateral of the somite is formed a dermomyotomes, a dense tissue. The dermatome form the derm and the subcutaneous tissue of the skin, while myotomes form the muscles of the back. Somite consists of the two parts, the ventral and the middle part of the somite, loose tissue, sclerotomes, later forming the fibroblast, chondroblast, and the osteoblast. Loose tissue surrounded the neural tubes and notochord, while the dorsal and the lateral of the somite is formed dermomyotomes, dense tissue. You can see dense tissue. Dermatomes forming the derms and the subcutaneous tissue of the skin, while myotomes forms the muscles of the back. Also in these sections, the cross sections or transverse sections, you can see a sclerotome, and this is the dermomyotome. A dermomyotome is that the lateral dorsal and the lateral part of the somite, a sclerotome, the ventral and the middle part of the somite, a sclerotome, a loose tissue, and the dermomyotome is a dense tissue. Next slide, the sagittal sections, 72 hours in the chick embryo. And these sections show two parts of the somite, the ventral and the middle part of the somite, a loose tissue, sclerotomes surrounded the neural tubes and a notochord. The second part of the somite, a dense tissue, a dermomyotomes. Okay, sclerotomes surrounded the neural tubes, here the spinal cord and a notochord. Sclerotomes, loose tissue, while the dermomyotomes, dense tissue. Thank you.